Eh, Hola, muy buenas. Os damos la bienvenida y en primer lugar agradecer a Joaquín Dopazo por la charla que acabamos de escuchar, que ha sido muy interesante, y a Raúl. Y bueno, lo que toca ahora en esta sesión es dar paso a uno de nuestros colaboradores, pero esta charla la vamos a hacer en inglés porque el ponente no domina castellano. Entonces le voy a dar paso en inglés y luego si tenéis alguna duda, pues utilizar, os recuerdo que tenéis la caja de chat para mandar preguntitas, ¿vale? Entonces, the next speaker belongs to one of our collaborators, concretely lab clinics. He's GERARDAS DAMBRAUSKAS, and GERARDAS is Director of Commercial Operation from Swift Biosciences. GERARDAS, welcome to our Congress, and feel free to start whenever you want. Thank you.
Great. Good afternoon, everybody. Thank you for the introduction. I believe you can hear me now. So I believe and you see my screen. So thank you much for your time today. We work with Lab Clinics, who is our distributor for Swift Biosciences products in Spain. And I was kindly invited to give this presentation and to give the overview of what Swift tools, what Swift tools that make for the next generation sequencing library preparation. And this is my talk solely focusing on the on next generation sequencing, how we make the DNA or RNA into the library prep. And today, just before I go into details, to give you a quick snapshot of Swift. Swift originally was founded in 2010. We started commercializing and selling the library preparation kits in 2014. The most platforms that are compatible with our kits are Illumina, Ion Torrent, to some extent Pacific Biosciences. The company has been founded on a broad IP portfolio that is covering our intellectual rights in the products that we develop. We are ISO certified. We have a big operational facility. So here you see the labs that make the kits. This is the R&D side and the production side, and this is the support area. So the company is based in Michigan in Ann Arbor, and in Spain, again, we work with Lab Clinics, our distributor for Spain market. So just to give you an overview, a quick overview of the libraries or the our focus areas. So we are focusing on genome sequencing, and these are the associated library kits. Then exome sequencing, targeted sequencing, epigenetics, so bisulfite conversion and library preparation. Then into the RNA sequencing, and the unique product that we have compared to other competitors, it's the library normalization kit. So obviously, with all these products, I cannot give a short overview today. I only got another 20 minutes. So what I will try to focus, I'll try to focus on some unique products that we do have in our portfolio, and they are based on the single-strand DNA or single-strand RNA incorporation into the library for sequencing. So mainly, I'll just full focus on the RNA kits, the methyl kit that we have, and 1S kit. All these three kits here, you see they share the same approach, it's very similar chemistries by putting the single-stranded nucleic acid into the library. And I will finish up with the normalase kit that is used to normalize the libraries ready to be pulled and sequenced. So Swift has a unique, we call it XL NGS adaptase technology, where we take a single-stranded DNA and or RNA in this case. So basically, it's a single-stranded nucleic acid, and we would add the tail, which also contains the adapter. The adapter is used for the flow cell to attach to the sequencing flow cell, and we call it this adaptase. So it's a single-step reaction that would ligate to any fragment that is 40 bases or longer. And this will be the addition of the 8 base. This is an 8 base tail, which has the adapter, as I mentioned. And this adaptase technology is incorporated into our epigenetics or methylsec kit 1S RNA library preps. So going to our gold standard, and I will try to reveal why we have a gold standard Excel methylsec kit, and it's already received its trademark. So just a quick reminder of what the bisulfite sequencing is. So when you have a methylated cytosines, in this case, when you use the bisulfite conversion, the methylated cytosines do not change in bisulfite treated or the reference sequence. So they stay the same. The non-methylated cytosines, would change to uracil in the converted bisulfite treated. So then they became a T when you do the DNA or the PCR on the converted sequence. So that's how you could distinguish the non-methylated ones or the methylated ones which do not change to the in a comparison to the targeted sequence. So for this we use the adaptase that is followed or that is preceded by the double-stranded DNA bisulfite conversion, as we've seen, and then you end up with a single-stranded. So we make a bisulfite conversion by itself breaks the DNA to shorter or longer pieces, and then the adaptase, which is in the kit, you ligate this molecule back to a single-stranded DNA, and then you create the library. I won't go into details into the library, but what I wanted to just to say that Swift, in comparison to other, especially homebrew methods, where the library is made before the bisulfite conversion, and basically what it does is that when you do the bisulfite conversion on already made library, this is very important. You basically break some molecules of your library, because your library must contain both adapters at both ends that are incorporated during the library construction process. So you may end up with some of the library that will have only one adapter, and that adapter obviously will not be sequenced in a downstream. So Swift has a unique approach to the market, and we are maintaining the uniqueness of the library in comparison to some other kits where you will lose some amounts of the library. So we can go to a very low inputs down to 100 picogram. So the range is quite wide. And we had the very successful publication comparing Swift libraries to Illumina and QIAGEN approaches in last year in Nature Scientific Reports. You could see them here, and the green color indicates that we scored higher than the competitor in Q30 fraction, as an example, having the lowest low quality basis, reproducible main insert size, less of adapter sequenced in the in the end sequencing data. Um, Lowest amount of read duplicates, the highest align rates. So truly, Swift methyl-sec kit stood out across the competition in that paper. So I already heard a little bit, although my Spanish is very limited, that there was a nice talk about the single cell application. Now, I didn't get a lot of details, but we also do have, as a company, a single cell methyl sec. We call it SNMC sec, developed with Salk Institute with Joe Ecker Lab. Joe Ecker Lab has been published. The first publication was in Science in 2017, where they looked at the single cell that you basically put into the library. So what they did, the single cells were from the brain cortex, was from human and mouse. The single nuclei or the single cells by nuclei staining were sorted onto the plate, and then they were bisulfite converted. The bisulfite conversion kit we use from Zymo, and we constantly have a streamlined process, bisulfite conversion from Zymo and methyl-sec from Swift, and then downstream application sequencing or sequence capture. So the cells were sorted on the plate. They were bisulfite converted. They ended up with a single-stranded DNA. You denature the DNA, and then you add the random primer. Random primer just to prime and incorporate one of the adapters. So you need to have an adapter into the primer. That was a little bit of a customized approach up to here. But then once you have the single-stranded DNA with the incorporated adapter, we come in and we bring the tail. So we're bringing another adapter. This becomes a truncated library with stubby adapters. Okay, so they are not full adapters. And then we bring the indices, and these indices were from Swift. We can bring to 384 now unique dual indexes or 768 combinatorial dual indexing. So you make a full-length, fully indexed library. So that was a success story from us that again yielded in the Science publication. And since then we had, I should say, another 2 or 3 publications in Science and Nature with this group. So they were able to process a single cell DNA methylomes from around 6,000 nuclei. So you could actually see that there is 3,000 mouse neurons, there are about 3,000 non-human cortex neurons. And they were able to identify the non-methylated and methylated patterns indicated here. And I think what they just a few important things, a few interesting things, that stronger cross-species conservation of regulatory elements in inhibitory neurons, and then in excitatory neurons between those species, mouse and human, and the patterns of the methylated, I believe, promoters were in sync or were very in a high correlation with when they did the RNA-seq library preps and RNA-seq results. So this was a really nice paper with a lot of details here. So another kit that we do have is Excel and GS1S Plus kit. It's based very similar on the same adaptase technology. You've seen the workflow. So it's a very similar. In this case, it's a single-stranded DNA fragment. It can be viral DNA as well. And where I'm going now is that we had this publication in 2016 where 1S kit was compared to tegmentation based, so, and rolling circle, or sorry, traditional multiple displacement amplification, so MDA method. So these two methods were looked at, and this was the reference sample from mock community with the known amount of species. Okay, and then when you look at the MDA or segmentation-based methods, the variety of the species that you identify, especially for MDA, very much differs from the mock community here. And this was the Swift 1S kit. And actually it very much mimics the species, with the exception of a few, what you actually find in the mock community. So we believe that this 1S kit gives a unique and very rich libraries compared to other methods indicated here. So because of that, we were very proud to be selected by the researchers who work for NASA. And they basically published this is in Science, that was last year. What they did, they looked at the twins, and one that went to the space and another one stayed on Earth. And what they did, they collected the specimens from both of these twins. Then they compare the microbiome using Swift 1S kit. So I could quote here that we were very most interested in being able to detect single-stranded DNA viruses as well as all other organisms as a part of our shotgun metagenome sequencing project. And this drove to the decision to use Swift 1S kit. So it was a multi-dimensional analysis. So there's a lot of parameters that were checked: microbiome, different cell counts, antibody counts, but 1S was used for microbiome for both of these twins. So I'm just switching a little bit now to the RNA kits that uses the same approach, this 1S adaptase tail. And just to give you some, just a quick snapshot, we random prime the RNA, and then we make a cDNA, so we don't focus on the poly-A in this case. There's no need to have a poly-A because sometimes it could be degraded. So we will random prime any really RNA, ribosomal depleted RNA, of course, if we don't want to sequence ribosomal RNA. Then we make it into cDNA, add the detail, and make the library, full-length library, just by indexing from the truncated library. And these are the wide range of the input. And we have, this is a longer protocol, 4.5 hours, but the input is very low, so we can go to the 100 picograms of the messenger RNA if we have. And then you can multiplex up to 768 libraries. And we have another kit, which is the rapid. We call it a rapid. So the input is slightly higher, but it's a very short protocol, and the time actually is the 3.5 hours here. What I was going to say here, that this kit is the fastest on the market compared to our competition. And we launched it about 6 months ago now, and we already have a huge success with these kits from Swift, and more into the richness of the libraries that I just wanted to show here. So this is the universal RNA input, and with 10 nanograms of input with Swift, if we are measuring so Swift is this blue bar, a Swift RNA is the red bars here, and this bar here is the supplier N, and supplier K in this case with 10 nanograms doesn't allow that low input. So what we are measuring here are the adapter dimers. So you're actually sequencing the adapter dimers here, so that would not be a good one. And the Swift stands out with very low input, having almost very little percentage of adapter dimers. And this is if we just put the messenger RNA. Again, we don't see that much of adapted dimers indicated here. So Swift, without going to details, work with the very degraded RNA as an input. If we're measuring on the RNA integration number score, you cannot assay it. It's very highly degraded RNA from, I believe this is from FFP kit, isolated with QIAGEN RNA is FFP kit. So the score of the arena number is so low that actually what we are doing, we are measuring the DV200. So all the area out after 200 nucleotides or 200 bases. So this area is measured. The arena number doesn't really give us any indication about any quality of the RNA here. And then we look into the library. So these are the libraries from both of those two samples, and they're not bad at all. So 100 nanograms was an input, and they were amplified because there was a very low input. So after you make the library, you use here 12 cycles of PCR to make it more detectable. And what we did here is that what we did then, so we made the libraries from those two samples, and then we did a capture experiment. So what? So rather than sequencing everything that you made into the library, we did here. We targeted with we have an exome panel. So we have an exome probes. So we basically use those probes to target and extract from the library the exonic or expressed regions. So in that case, you are doing like RNA, looking at the expression, RNA-seq, looking at the exomes that are expressed. So we capture those regions. I won't go into details, but this is a very elegant approach because you don't have to do the ribosomal depletion. So you take all the RNA, convert it into the library, fish out the exomes, and sequence. And a second thing is that you can multiplex more samples because you are enriching for the exons. Okay, so you can multiplex at the end into the pool more libraries. That makes your assay more cost effective. And then we hybridize. So so these are the reagents and the 100 nanograms of each library that was hybridized. We eluted the libraries. And this is how they looked after the elution, post capture, and then we did the number of genes that we detected, indicated here, and there was very highly reproducible between those two samples. We looked at high parameters. We looked at the transcripts detected, genes detected, the library complexity. And yeah, this is we calculated that you can probably scale down the sequencing by the factor of 4, the cost of sequencing by the factor of 4. Okay, so the percentage was 92% on target. So what we targeted on bait versus selected, so we captured about 92% what we targeted. So just a few slides on everyone's talking about SARS-CoV-2. Swift approach is sequencing the actual virus. We don't develop the qPCR kits. We go into the viral genome indicated here, and we go it by amplicon. So we are amplifying the regions of the interest. It could be and they are overlapping. So basically these amplicons are to some extent overlapping. So we can actually sequence very efficiently about 30 kb, about 99% of the SARS-CoV-2. And this is a very elegant amplicon-based approach. So what we do here is that a single-stranded RNA needs to be converted. We don't produce this kit, so we advise which kits that can be used into the cDNA synthesis. And once you get the cDNA synthesis, on cDNA we do the amplicon amplification and amplicon downstream library preparation. And interestingly enough, we had the first publication in PNAs this year, very recently in September, from Joachim Sagalas. Excuse me if I did not pronounce the name very well, but they looked at the SARS-CoV-2 infected, and this is a cross-species now jump from the owner or the family that got infected and they carried the virus, and they assayed the cat, which unfortunately had to be deceased because it had other problems. But what they did, they tested for SARS-CoV-2 and they found SARS-CoV-2 sequence by RT-PCR, just to prove that the virus was there, and they were looking at the actual viral sequence. But we already have that article that was successfully published, and SARS-CoV-2 RNA was only detected in nasal swab, nasal turbinate, so I believe it was nasopharyngeal and mesenteric lymph node. This was a very interesting finding as well. So that's, and then it was put into the sequencing, and they were figuring out, and they found that the actual sequence from the cat was the same as from the owner. And just to briefly finish it up, we do have a Swift normalase kit that would normalize your libraries prior your pooling. So researchers do spend quite a lot of time when they make the libraries. They have to pool those libraries in order to sequence them together to bring the costs down. And the reason is that you want each library to have equimolar concentration, or each library be represented equally in the final pool. So you're doing qPCR, quantitative PCR, or qubit or bioanalyzer to identify the library concentration. Then you go back to the library and you pool the and you dilute the libraries. So more concentrated libraries get more diluted, and then you put it into the pool. So what do we do here? We have this normalase kit that takes away all that normalization step. And rather than doing the conventional workflow, you just use a normalase. And the way it works is that you have your different libraries, you do a normalase PCR. It's a one short 3 cycles, minimum 3 cycles of PCR. And then you put libraries into the normalase 1. This is an enzymatic step. So basically in a separate tube, you add normalase 1. You keep it for 15 minutes at 37 degrees incubation. Then you pool all those libraries into one pool, and then you add normalase 2, indicated here. And then you have in your library, your final pool, the library that is at a certain concentration, in this case at 4 nanomolar. So in your normalase 1 will mark a certain concentration of your library separately. In the pool, normalase 2 will degrade the excess of your library, and you will only end up with the libraries that at 4 nanomolar final. So this is how it works in actual library construction diagram. So normalase 1, normalase 2, and you end up with a single-stranded normalized library pool at 4 nanomolars as an example. So by doing the quantitative PCR and using the quantitative PCR results, when you're actually getting the reads out of your sequencer and you are demultiplexing, deconvoluting the reads, and you are assigning each read to each sample, you can actually see that some of the libraries get more reads and some of the libraries get lower reads, and the coefficient of variability, or the variation between the libraries, is about 20%. With normalase, our customers achieve now below 5% variability, which means that each library gives a very consistent in the pool library reads. So we had this customer at Tony Brooks at UCL here in the UK. I'm based in the UK. And our customer, he did starting concentrations of each sample. You can actually see here in red, and here starting nanomolar concentrations. And the samples were above 10 nanomolar. So this is the requirement to go into the normalase. You have to have 10 nanomolar, sorry, 12 nanomolar of your library. If you don't have the 12 nanomolar of the library, the other alternative is to have 6 nanomolar of your library. And then you go into a sequencer, and so in the normalase will equalize those libraries. And then when you look at the reads in blue here, the coefficient of variation in his first run was less than 10%. So imagine if you have to do 96 libraries, there's a lot of qPCR, there's a lot of playing on the plates. So if you have 96 and you're doing normalase, you add with multi-channel pipette, normalase 1, you incubate it. Then with multi-channel pipette, you collect everything in one tube, you incubate, and then you have, and you do a cleanup, and then you put it into the sequencer. That's how simple the normalase is. And then he did exome run. This is again from, not necessarily from Swift Biosciences, they did it from NAB in this case, and the coefficient of the variation of the exons were 3.7. So there was not much over sequenced samples nor under sequenced. And then the last slide is that it turns out that normalase mitigates the index hopping or the index misassignment. That mainly happens on the pattern flow cells. A high-sec, NovaSec has the pattern flow cell, and with normalase, the index misassignment is compared to no normalase goes from 0.3% down to less than 0.05%, and this is what we saw. And this is the courtesy of Tony Brooks, our customer, who also sees the normalase hopping rate just below 0.11%. So it's very rare phenomena, but it can then mix your samples index hopping. So with this in mind, I believe I presented some of the tools that Swift is heavily working, and we're having some success. And thank you very much for listening, and the organizers, thank you for the invitation.
Thank you very much, Gerardas. We are a little tight to the time, but you have one question. The first one is from David Rubio Mangas, and he's asking that, is the methylation kit compatible with highly as FPPE and liquid biopsy?
Yes, absolutely. And we do have some publications. Yes, it is compatible with FFPE and liquid biopsy, especially the cell-free DNA. We just had the publication on a cell-free DNA looking into prostate metastasis and finding the signature in plasma. So using our methyl-sec kit, yes, it is compatible, fully compatible.
Okay, and you have another question from Miguel.
Sorry, you're breaking. You just broke.
Any of the... sorry. There is another question from Ruth, and is, will any of the library preps kits suitable for the use with fax samples with few cells?
With fax samples, yes, absolutely. So our kit, we call it a single cell adaptase module. We do have it, and some of it was shown today in my presentation. And the minimum of cells, you probably looking into about 20 cells that a single cell adaptase module would work, and you would get sufficient DNA out of it. Yes.
Okay, and then thank you very much, Gerardas. Thank you for presenting us your products. We have to say that we are very grateful for that clinic's cooperation. Well, that's all. And now, well, I will speak in Spanish now.
Thank you very much. Great, thank you.
Thank you very much, Gerardas. Es el turno de la cuarta sesión, la sesión plenaria que corresponde a Mario Delgado y va a ser presentada por Alba Rodríguez. Cuando queráis, muchísimas gracias a todos por asistir. Un saludo.